back here involved in like balance and motor controls. And here are a bunch of the different cortex, cortical regions, all right? There's this sulcus, which is this sort of gap here. Right behind it, this is somatosensory. This is where you feel, your touch senses are. Right in front of it, by the way, not drawn in here, is a bunch of stuff having to do with motor control, controlling your muscles. Auditory cortex in here. And then this massive piece of stuff, and it doesn't look quite so massive in here, but it actually folds inside your brain. We're going to have to unfold your brain in a minute. That's all visual cortex, and that's all the processing of visual information. If we unfold your brain, okay, which is generally painful, all right, what we have to do is we have to sort of take all this crinkled stuff and then open it up and layer it. What you would see is something that looks like this, and bear with me here, okay? What's happened here is this here is your eyeball right there. This back area is V1. They're showing this is on the inside. This is on the outside. Here's your nose here. This is this. These are your visual cortex areas here. And if I were to peel it out, so take this brain and flatten it out, okay, you would get a map that looks something like this. So this big area here is V1. That's this area back in here. It wraps around. And then there are these other visual areas. Anything starting with a V is a visual area. And then it goes all the way up. And here's that. So this is wrapping around this whole body, right? Here's that somatosensory, the feeling. Here's your motor cortex. Whole bunch of these other areas. Okay? This is what you would see. All right? But of course, this is just a drawing. Now I want to show you a picture that's kind of cool and kind of gross at the same time. All right? And here's what we're going to do. And again, PETA folks, turn this off. All right. You take an animal, living animal, you immobilize its eye by giving an injection that prevents the eye muscles from moving the eye. Okay? You put up a flashing pattern that's in like some rings that alternates sort of bright and dark spots, and you turn that on. But remember, the eye can't move, so the eye is fixed at the same point. You then inject essentially a type of radioactive substance into the bloodstream that binds with sort of the oxygenated red blood cells. And the more cells work, the more oxygen they consume. Okay? So you can measure, so this was like one of the first ways of measuring the activity of the blood cell, of the brain cells by, you have them do this work for a while this radioactive tracer is connected to the red blood cells and oxygen. And what happens is the blood cells that are doing a lot of work build up a lot of this radioactive material. But of course, you have to get a picture of that radioactive material, and it's a very thin little bit of material in a very thin part of the brain. So what you have to do is what they refer to as euthanize the animal. So the animal is killed. You then very carefully unpeel its brain. And you can take a radiograph. You can actually measure, you can think of it as simply as putting that brain tissue next to a photographic plate, and the radioactivity makes an image. Okay? There's a little more to it than that. When you do that, you get this remarkable picture, and this one comes from 1988. So even though this is sort of very fundamental work, it's not all that old, okay? And here is what the animal was looking at, okay? It's macaque, all right? So here you can see the visual stimulus the animal was looking at, and here is that picture of its brain. And what's so cool is that this structure is preserved. This is what's referred to as retinotopic mapping, is preserved within the cortex. Okay? So when you move a little bit in the retina, you move a bit in the cortex. Now, what's also really cool is that it is not one-to-one in terms of how much cortical area is devoted to how much of the retina. In fact, the way it's drawn out is sometimes referred to log polar, etc. And again, this is where we've unfolded your visual field. And you see that like this little area, 1, 2, 3, 4, is looking at a lot on the visual cortex. And then this area out here, okay, is much, much smaller. So you use more brain real estate for that tiny little area in the middle of your visual field than you use real estate for the bigger areas out here. Which also, I mean, yes, it makes sense because that's where all the processing needs to be. But the fact that you can actually find this in the actual tissue is, in some sense, remarkable. The other way that we can do certain types of understanding what's going on in the brain is with what's called physiological measurements. And so again, using animals, typically what you'll have is you have a screen where certain things are being displayed, an animal whose head is restrained so it's looking in a particular direction, and there's actually an electrode recording from the neurons in the brain. You hook that up to an amplifier. In the old days, you'd look at it on an oscilloscope, and actually you would just connect it to a speaker and you'd listen. And more clicks meant you were getting more response, and you'd have to write it down and report it. Then you had an oscilloscope that had traces. Now, of course, it's all computerized. All right. You've seen this picture before. This is a picture of what it looks like for a microelectrode to be recording from a neuron. And up on top, this is what the actual voltages look like, these spikes. And so then down here, you have something that just sort of counts the spikes that go across. So one of the first areas that people want to look is what was called V1. Back in the old days, it was actually called Area 17, I think, but that sounded too much like UFOs or something. I don't know. It's V1, the first visual area. And when you record from the first visual area, and here's an example, the way you get a monkey to look in a particular place is you give him juice whenever he pushes a button when something happens, and so he'll attend very carefully there, and it happens, he pushes the right button, he gets juice, and apparently monkeys really like apple juice. But while you're doing that, you're doing other things to the visual field. And one of the things they've found is that there are cells that if you put up a bar, if you put the bar, let's say, horizontally, you get hardly any reaction at all. In fact, put it up at a 45 degree, still hardly any reaction. Put it up vertically in this case, and you get a tremendous amount. And by the way, again, this is the part where it's off, this is the part where it's on, this is the part where it's off. Now, for this particular trace, I don't know if the bar was moving at all or just being flashed up there. The movement I'll show you in just a minute. But the idea is that within V1, the first visual area of cortex, there are cells that are extremely sensitive to the orientation of essentially edges. Remember we did oriented edge detection? So this feels an awful lot like oriented edge detection. It would be an oversimplification to say that's what it's doing, but it's related. If you recall, we also talked about motion. Well, it turns out in V1, you also get directional selectivity, all right? So here we have a cell, and these, of course, are the spike trains. And this particular cell, if you have a bar that's tilted a little bit and moving upwards, you get a small amount of motion. If you have a bar that's here on this 45-degree angle and you moved it upwards, you got a lot of spikes. What's remarkable is when you move it down, you got no spikes at all, or even less than resting state spikes. So not only do we have orientation selectivity, we have movement selectivity. And you can actually make a pretty cool plot of how this distributes in the brain. And so this is essentially, I guess this is your brain on drugs, because, I don't know, anyway, it's all colored. But the idea is that each of these colors represents these different orientations, and you can get these mappings, and you can see that it's not randomly scattered individual points. There are regions, all right? And in fact, these are referred to as hypercolumns in the sense of that little tissue areas within the brain will all be associated with a particular movement. Now, remember, it was retinatopic, so this area is all from one part of the retina? Sure, but within that area, there'll be little subparts that are for verticals and oriented edges like that, including different kinds of motion. So those were the kinds of things that people saw in these first visual areas. There was a group of neurophysiologists, Hubel and Wiesel at Harvard, who I think did work in cats originally, where they first looked at these some of these cells. They also looked at things called XY cells having a steady state in motion, and they won the Nobel Prize for this work. Okay, so this idea of trying to understand how the visual cortex was processing the visual signal was really, in fact, that Nobel Prize was announced when I was in grad school, so it was in the 80s at some point. So that gives you an idea of sort of the recency of some of this work. So now that we have bars and edges in V1, we can go upstairs a little bit to V2. And people do all sorts of work of looking at different types of patterns that cause different effects. And so some of them patterns were based upon like little gratings and shapes and swirls and things like that. And sometimes they were just on these types of edges and things. And it's by David Van Essen's lab, and they're very well-known neurophysiologist. But when you look at things like this, it's a little difficult to understand, exactly how this is moving upstream in the computational world. We understand the need for edges, and then you can maybe make some argument about circles and or curved parts. One might say that, what I'm seeing is stuff that may be dealing with some types of intersections, those kinds of things. But the neurophysiologists, they keep looking to understand what sorts of images or low-level features will cause cells to respond. Going upstairs even a little bit further, there's an area called the infratemporal cortex, and Tanaka did a lot of work on looking at different kinds of features that seem to have cells that have a strong response to these and not other kinds of things. And the idea was that maybe these are where cells are starting to put things together, okay? And I guess one of the most well-known examples actually came, it was earlier, and the dates go different. There was this so-called hand neuron, okay? And what this was is this is a neuron that when you showed it a picture of a hand, it would fire a lot. And the front of a hand, the back of a hand. And remember, these are kind of like line drawings of hands, and even this one and that one, so different size scale. But what was interesting is if you had an image that just had these little spokes there, it doesn't look like a hand to me, doesn't look like a hand to you. Well, obviously, it didn't look like a hand to this cell either. And the idea that this was a cell. Now, you might wonder what this picture of this person looking like is. Well, there was this whole conversation of what was called, did we find the grandmother cell? That is, can you find a cell that just fires when it sees my grandmother? And I think this was just a play on that. But on the one hand, it was kind of cool that you would find a single cell that was responding to, say, just hands. On the other hand, we know for a lot of reasons that it can't be as simple as, there's a one-for-one mapping between concepts and cells, because there's all sorts of reasons why that would be a bad design. But we're getting at this idea that what's being processed is no longer just very simple features, but something like, this, which is obviously a collection of features in a useful way. To further point that out, here's two pictures of what you and I would say are approximately same-sized silhouette of a rhinoceros and a lion. And these look sort of similar in terms of where the stuff is, okay? But of course, they're different animals. And what's nice is that in V1, these trigger similar cells. So it's like cells that are responsive to the edges in this location, because the edges are sort of in the same spot. But of course, they respond differently in IT. IT is infratemporal cortex. The idea is that maybe in the cortex is a cell that understands something about rhinoceros, so it won't respond when it sees a lion. And in fact, you can further confirm that by showing it, if you find a cell that sees lions but not rhinoceros, now you show it two different pictures. One that's a line drawing of a lion, and the other one a much smaller filled-in silhouette of a lion. These objects in some sense look different at the picture level, but they are the same thing. And unlike the previous pictures where they are different things but similar at the picture level, these respond very differently in V1, the early processing, and then there are the same in the infratemporal cortex. So the idea being that higher up are cells in the vision system that understand something about the appearance of object types. At least that's that's sort of the story. But what if you want to actually know what's going on inside human brains? And presuming these are people you actually like. So how would you measure what's going on in their brain? Well, you're not going to stick electrodes in their brain. What you're going to do is something called fMRI, okay, or functional magnetic resonance imaging. So many of you probably have had an MRI, or an MRI uses strong magnetic fields to sort of mess around with electrons in order to be able to get differential density measurements. Well, you can do something cool also. You can use a dye contrast that the magnetic fields perturb differently depending upon how much oxygen is in that part of the blood. So just like we did before with that experiment with the monkeys where you injected sort of that radioactive isotope connected to oxygen, now what we can do is we can have you have dye contrast, go into an fMRI, have you look at things and see what parts of your brain are lighting up. All right. So here's an example, sort of notionally, of an fMRI activation. So this is, of course, obviously a reconstruction based upon the density. And here you can see the activity that's being done. This is a three-dimensional reconstruction.